I wanted to say a couple of words because it leads into Matt's presentation in sort of a unique way. And so I was reflecting this afternoon about things that I haven't chatted about in a little while and why it's important to talk about these things at a public lecture. By the way, I'm Eileen Anderson. I direct the Stem Cell Research Center here. And this is a slide that I used to show in our pre-COVID times when we would really pack this room once a month on Tuesdays with a variety of high school students and community college students and individuals from the community and of course our UCI researchers. And it's something that a lot of people don't realize. So one of my huge, like the things that just gets under my skin is when you see those pharmaceutical company commercials that have people in white coats in beautiful environments and they talk about what an amazing job they did bringing Celebrex or whatever it is to being a therapy. And the reality is that that was probably almost entirely funded by your tax dollars and the National Institutes of Health. And then companies pick up on those pieces and move them forward to the final stages of translation. That's really important. It's incredibly important work. But none of it would happen without the sorts of things that happen in this building. And I think that message gets lost a lot. So this is a study that came out, I think in 2017 or 2018, that just looked at 2 years of research at public institutions, which generated over 13,000 patent applications. And in fact, it reviewed how many new molecular entities, so new drugs that came to market in this same period, and all of them could be traced to federal or state funding for basic science research. So this is the driver of how things get through to clinical translation. What happens in this building and other buildings on this campus are absolutely critical and essential for that. And I'm going to show you a couple of examples of that. Just in the last 10 years, these are the spin-off companies that have come out of people that worked in this building alone. And I thought it apt to highlight that tonight again, because one of the people on here, again, arguing about PowerPoint versus Keynote, which is a much better program, Matt, are these three individuals, one of which, of course, is Dr. Blurton-Jones, right? And so he's in the process right now, and some of, I think, what he's going to tell you about tonight is the discovery stage that leads into translational work that we do in our labs and on into things like creating spin-off companies that have the chance to go on and change how cell and gene therapies and regenerative medicine as a whole impacts all of our lives on a day-to-day basis. And that is why it's not just about innovation, but it's what I'm calling here for the purpose of this slide, discovery, basic science discovery at work is this process where we spin out into the community and move things forward to clinical trial. So towards that end, I thought I would also highlight a couple of the things that we have here at the center that are important for that actual testing part in the clinical scenario. So we have, some of you have heard me talk about over the last 2 years, a CIRM-funded Alpha Clinic. That's an $8 million grant that also School of Medicine and College of Health Sciences supports. That's for the actual translation piece. So how do we get out of the bench into the bedside? At some point, we have to conduct clinical trials. That is an FDA, Food and Drug Administration, authorized process. And we have all of the services here to be able to support that. And in fact, this is our trajectory over just the last 4 years in terms of the number of clinical trials at UCI that are being run in the Stem Cell Research Center for various clinical indications. So this year, we're on track for over 60 clinical trials that will run through our Alpha Clinic here, directed by Daniela Boda. Another critical piece is how do you make those cell and gene therapies? Well, you make them in a place called a good manufacturing practice facility or a GMP facility. And this has been, oh my gosh, probably going back to 2016 or 2017 in terms of the planning stages at UCI. But just in January of this year, we got our license for this facility. So School of Medicine in the main campus invested over $13 million. But at the end of that, we have a 7-room manufacturing facility that received our California Department of Public Health license technically on February 16th, but the inspection was on January 8th. And that means that we can actually not just do the clinical trial piece, but the manufacture of the therapeutics that will be delivered in clinical trials here. And in fact, we've run our first project through here. One of the next ones that we anticipate running has to do actually with one of our own spin-offs, J-Site, which is a therapy for retinitis pigmentosa. And the GMP facility here and Gavin Herbert Eye Institute and the Alpha Clinic, we expect to all be involved in their phase 3 trial for that indication. So super excited about that. In that regard, I just want to highlight for everyone here, on May 22nd, we're having our sort of ribbon cutting of that facility. We'll be sending out some information from the Stem Cell Research Center, SOWA School of Medicine. Everyone is welcome to attend. Everyone can't do a tour, but we will have some limited tours that happen, and we'll have some nice presentations about what exactly goes on in the GMP facility. So please mark that on your calendars. And with that, I want to welcome Matt to give his talk this evening and to please come up. So ruthlessly, I've adopted our founding director's strategy of introductions, and that's to ask questions. I really like it a lot. So Matt, how did you come to be at UCI?
Wow. So I did my PhD down at UC San Diego, and my wife and I were starting to think about where we were going to move, and she was about to start a residency up here, and so I was commuting for a little while. So it gets a little old to commute to San Diego, and so I started looking into the various faculty here at UC Irvine and found some amazing neuroscientists studying Alzheimer's disease, which is what I was interested in. So I applied for some postdocs, and that's how I started.
Fun fact, Matt and I actually overlapped for a brief period of time in Carl Koppmann's lab on campus.
Right.
So, yeah. So I'm super excited for your talk. I think there are a lot of people here that want to hear what you have to say about this next generation thinking about cell therapeutics. Matt's going to tell us about a specialized immune cell in the brain called microglia and how that might be able to be targeted or purposed. There you go.
That's right. Okay. Thank you so much, Eileen, and thank you all for coming. It's a great showing. I'm excited to tell you a bit about some of the work we've been doing in my lab over the last several years, trying to study these cells called microglia. And by the time you leave tonight, you're going to all be experts in microglia and know everything about what they can do and what cool cells they are. As Eileen mentioned, I recently started a company, and one of the things one needs to do when you start a company is you have to put your disclosures here to make it very clear that, yes, I started a company with some friends and colleagues, and it is building off of some of the work that I'm showing you here, but all of the work I'm going to show you is done here at UC Irvine, not by the company. So just a brief outline of what I'm going to tell you about today. We're going to be talking quite a lot about Alzheimer's disease, and that's a disease I've been studying for many, many years since graduate school, and continue to study. And I'm sure many of you are familiar with Alzheimer's disease, but we'll get into some details about what's underlying this disease, why we don't have treatments that are very effective yet. And then I'm going to tell you about how we're using stem cell-derived microglia to study Alzheimer's disease, and specifically to study some of the disease risk genes that have recently been identified. And then we'll switch gears and tell you about some very new work on whether we might be able to use these cells, microglia, for development of new therapies for potentially Alzheimer's disease, but potentially many other neurological diseases. So sadly, I think everyone is familiar with Alzheimer's disease. You probably all know someone who's affected by this disease. And that's because it's the most common form of age-related dementia. And it gradually destroys a person's ability to learn, reason, carry out daily activities, and of course, dramatically affects memory. And we know that when a patient passes away from advanced Alzheimer's disease, there's significant atrophy of the brain. So on your right side is a sort of a cross-section of half of a brain of a patient who had Alzheimer's disease, and on the left side is an age-matched control. So Alzheimer's is influenced by age, but it is not a foregone conclusion with age. It is a disease process. But you can see there's really dramatic loss of this tissue, especially in the cortex here. You see this big hole here that should, It should be a small little hole where cerebral spinal fluid is flowing. And then you see this other sort of missing piece of tissue here that should look like that. That's an area called the hippocampus, which we know is very important for learning and memory. So it kind of makes sense that that's so dramatically affected in this disease. Now, unfortunately, the number of patients who are going to be affected by Alzheimer's disease in our country and worldwide is going to continue to increase. And this is largely driven by demographics. So you can see this is split by age, and here we are around 2024 here. We're a little about 6.5 million people with Alzheimer's disease right now in the US. And the estimate is that by 2050, we'll be getting up to about 14 million. And it's really because of this purple bracket here and maybe a little bit the yellow bracket. This is split by age. So our population is aging. The baby boomer generation is getting to that age where they're starting to approach a greater risk of developing Alzheimer's disease. And so as our population ages, so too does the number of patients being affected by Alzheimer's disease. And so it's already a very big health problem for our country, but it's only going to continue to grow. And so there's a real need to find better ways to try and treat and slow this disease. So we've known quite a lot about the pathology for some of it for over 100 years. So Alois Alzheimer, the German neurologist who the disease is named after, first described 2 hallmark pathologies. And he didn't know the details biochemically of what they were made of, but he looked under his microscope and he saw these strange things, these amorphous aggregates that were outside of the cells, but in the brains of these patients. And we now know them as beta amyloid plaques. So many years later, it was discovered that these are made up of a small protein called beta amyloid that starts to aggregate and becomes insoluble and accumulates extracellularly in the brains of patients with Alzheimer's disease. Now, Alzheimer himself also described another pathology that are called neurofibrillary tangles, and these are also an aggregation of a protein that occurs actually within the neurons, within the brain cells. In this case, it's a protein called tau. It has a normal function of sort of maintaining the skeleton of the cell, but it also starts to get aggregated, phosphorylated, and accumulate, and eventually causes disruption of the function of the neurons and even the death of those neurons. Now, other things that we can see in Alzheimer's disease brains include this image here, where now we've changed the color of the plaques to be blue. So in the center here is one amyloid plaque. And around it, you can probably make out there's several cells, little cell bodies here. Each of those cells in red is a microglia. And microglia are essentially the immune cell of the brain. And they seem to be surrounding the plaques, and we see that very consistently in Alzheimer's patient brains. You may even note there's a tiny little blue dot in this one here. There's some evidence that they might be able to eat some of that amyloid. In fact, microglia being an immune cell, one of their jobs is to clear up the mess by eating, by a process called phagocytosis. And so they may be trying to clear that amyloid through that process. Now, in addition to this, we have then neuronal loss and the loss of connections between the neurons. So synapses that are these connections that occur between the neurons that allow us to have complex thought. So that neuronal loss and synapse loss is ultimately what drives the cognitive impairment, but it's driven by all of these different factors interacting at different times and in different ways. So we do know that there's a number of things that can influence Alzheimer's risk, and of course the biggest one is advancing age. Alzheimer's is an age-related disease. The older you get, the more likely you are to develop Alzheimer's disease. We also know that low education level can contribute to Alzheimer's disease, and this doesn't just mean formal education. This means not challenging your brain. So for example, the fact that you guys showed up tonight to listen to this, you're learning, that means you're protecting yourself against Alzheimer's. So kudos to you. You've probably also heard about how previous head trauma can impact and influence the risk of Alzheimer's disease. And there's a related disease, one that Brian Cummings here studies, called chronic traumatic encephalopathy, that exhibits many of the same pathologies that we see in Alzheimer's brains. And so you can see this overlap between these two pathologies. Diet, this is another disease where a healthy diet is a good thing. There's lots of studies suggesting a Mediterranean diet is protective for Alzheimer's disease. Likewise, exercise is beneficial. So we don't want to be couch potatoes like that recent Super Bowl commercial. And then, of course, like most diseases, there's a genetic component. So family history of dementia can contribute to your chances of developing Alzheimer's. And I just want to comment briefly on this term dementia, because we often use this interchangeably, dementia and Alzheimer's. They're not really the same thing. You can think of dementia as an umbrella term. So under dementia are various causes of dementia. Alzheimer's is the biggest cause of dementia, the most prevalent cause of dementia, but there's other forms of dementia, things like frontotemporal dementia or Lewy body dementia. So when we say family history of dementia, this is partly because when you go back a generation or two, physicians, all of us weren't very aware of the difference between these different dementias. And so sometimes someone would be diagnosed with a different dementia or just told they were senile without knowing what was driving that disease. So the field has learned a lot from some very rare forms of Alzheimer's disease that we call familial Alzheimer's disease. Really, these provided the first clues as to what was going on and how this disease was progressing. And it turns out that Alois Alzheimer's himself, the first patient that he reported and described, shown here, August D., who was only 54 years old. I think she died at 57. And this is, during one of her lucid states, a quote of hers, "I've lost myself." So that's a very young age to be developing Alzheimer's disease. And as I said, not surprisingly, they found out that she was carrying a mutation that is associated with these rare familial forms of Alzheimer's disease. And that's one of the ways we know it's this familial form, is they tend to be very early onset. So patients will develop disease sometimes in their 40s or 50s. The earliest case I'm aware of is 19, so it can be quite aggressive and quite early. Now, Alzheimer's got on his fancy microscope at the time and did beautiful drawings like the one on the right here, and then here from a colleague of his, Oscar Fisher. So in Fisher's drawings, we can see one of these amyloid plaques here with these funny little bulbous things attached to them that we now call dystrophic neurites. They're actually disrupted neuronal processes. And then Alzheimer's did these beautiful pictures of the tangles. Now we can fast forward 120 years and we can buy a $300,000 microscope and get slightly more colorful pictures perhaps, but essentially showing the same thing. So these is some examples of amyloid plaques in blue and some of these dystrophic neurites around these neuritic plaques. And here's a couple images of tangles that I took when I was a postdoc. So we are still looking at these pathologies and still focusing on these pathologies and trying to understand how they're driving the disease. And we've learned a ton. I don't want to belittle the progress that was made here, but there is still more to learn in terms of how these diseases, how these pathologies relate, what's driving them, and how they are contributing to cognitive loss. That said, as I mentioned, there's these familial forms, and it makes sense that these were recognized first. They are dominantly inherited. So if one parent developed this disease, the kids had a 50% chance of developing it. And so you'd see the development of this disease at a very young age. That gave us what we would call pedigrees, these family histories. You could very easily trace them. And as people started to investigate genetics, we were able to pinpoint the genes and find those genes. And they discovered 3 genes that are sort of abbreviated here. APP, which stands for amyloid precursor protein. So as the name suggests, it's a protein from which amyloid is derived. It gets cut up into smaller fragments to produce amyloid. And then these other ones, presenilin 1, presenilin 2, well, those are catchy names because they were associated with Alzheimer's. But what we know they do is actually cut that amyloid precursor protein to liberate and release the amyloid. So all 3 of those genes were pointing towards amyloid, that amyloid was driving this disease. So all of those mutations have been discovered, and they found many different types of mutations in those 3 genes. They all either increase the amount of amyloid or they increase a more toxic aggregation-prone form of amyloid. And so that led a lot of us to focus, the field as a whole, to focus on these mutations. We saw the plaques, we saw the amyloid in the brain, we saw these examples driving this disease, and kind of ignore this 95% for a while. It led to something called the amyloid cascade hypothesis, which makes a lot of sense, and there's still debate about this, and I would say still probably 95% of Alzheimer's researchers think this is roughly correct, but it was overly simple. So what the amyloid cascade hypothesis suggests is, and as I said, it was informed by these rare familial cases, it hypothesized, it predicts that the accumulation of beta amyloid is the initiating trigger that drives the development of both sporadic and familial Alzheimer's disease, right? So it was based on familial, but it kind of assumed, well, they look the same. We look under the microscope and the brains look very similar, so maybe it's driving both of these. And the difference, though, is how that amyloid might be generated. So in the case of the familial, it seems to be overproduction or sometimes missed metabolism. And more we're getting evidence that for the rest of the cases, there's problems with the clearance, the ability to get rid of amyloid. So as I said, although this was based on these earlier studies, it continues to hold up that this may be what's driving the disease. But we're going to talk about, well, if we've known about this for so long, why haven't we found a treatment for Alzheimer's by going after this amyloid? So essentially inspired by this idea, pretty much every biotech pharmaceutical company tried to develop drugs to go after it. And you probably have all come across, these sad stories of failed trials for Alzheimer's disease. Many companies went bankrupt, other ones completely shut down their program. Unfortunately, some companies continue to work on this, trying to push forward and figure out how do we treat this disease. And you may have heard about last year that 2 new drugs were approved for Alzheimer's disease. And this was the first time in a long time that any drugs were approved with Alzheimer's. And they were heralded as what we call disease modifying, the idea being that they can go after that amyloid and reduce it. And both of these drugs essentially work as antibodies. They're antibodies against amyloid that try to bind up the amyloid and have your immune system help to clear that amyloid. And from these clinical trials, they would do things like PET scans. So there's a type of PET scan depicted here where you can actually get an idea for the amount of these amyloid plaques in a patient's brain. So the brighter the signal, the more amyloid in this patient. Here was a patient who was given placebo for 18 months. Here was the patient given this drug for 18 months. And you can see there's still plenty of signal, but there's less in that patient. And it was significantly less. There was a reduction in amyloid in those patients. So that was promising. Obviously, for patients, what they really care about, and their loved ones really care about, is this actually helping with their cognitive impairments? And that was one of the kind of big breakthroughs compared to those past studies. Is yes, it led to a statistically significant slowing of cognitive decline. And this is the data right here, the sort of key data upon which this drug was approved. So this is measuring the decline in cognition using a sort of a set of tests combined to get an average score. And you can see 2 lines here. The black line is the patients who for 18 months were taking a control, placebo. And the green line is the patients taking this drug for 18 months. And so you can see over the course of that 18 months, they start to diverge, right? And it becomes very, very significant. The problem is the scale here. So there's actually still quite a lot of debate about whether these drugs provide meaningful slowing of cognitive decline. And that's because over this 18-month trial, it slowed decline. If you look at the distance here, it's about 0.45 on this cognitive score that's very commonly used. And yet most clinicians who see Alzheimer's patients will tell you that a patient and or their caregiver really can't tell if there's a difference unless it's over about a 1-point change. So would they recognize this difference? Probably not. the hope is that maybe over extended periods of time, we'll get more separation and this will start to show benefits. But what I want to get across here, and I don't want to be too depressing here, but this is not a silver bullet. This is not a cure for Alzheimer's disease. It's a Band-Aid that perhaps with time, perhaps with starting earlier, we might see more benefits from these drugs, but we still need to do a lot of work to try and figure out how this disease is continuing and other ways to treat this disease. So one of the potential explanations for this is kind of depicted here. So we've learned a lot from these PET scanning approaches or measuring what we call biomarkers, so levels of amyloid or tau in the blood or in the cerebrospinal fluid. And over the last 20 years or so, we've been able to build these sort of curves that tell us a bit about how this disease proceeds. And so on the left is starting at the beginning, normal, cognitively unimpaired, and on the right is advanced Alzheimer's dementia. And so you can see the black line is actually the amyloid, and it's coming up quite quickly. The tau pathology comes up a bit later, often about 10 years later. The green is structural changes in the brain, so loss of brain tissue that you can see by MRI. And then the red is cognitive impairment, actually being able to see a difference in the way someone's memory or behavior is. So when we look at these curves, it's telling us that the amyloid is occurring quite a bit before that cognitive impairment, and it's in fact 10 to 15 years before cognitive impairment. So it's a very slow process. And the problem is these clinical trials are being initiated here. We're finding patients who have cognitive symptoms, asking them if they want to be enrolled in a trial, splitting them between placebo and treatment groups. That's when they get started, and amyloid's already pretty high up, right? And so I think, at least my thinking on this, and I think a lot of people's thinking on this, is that this analogy tells us what's happening. So here's this very heroic guy who the apartment next door is on fire, and he's taking a little kitchen bucket and trying to put out that fire. But even if he had a massive fire hose and put out that fire, there's a lot of damage done to that apartment. So yes, these drugs, they're a bit like the bucket. They can put out the fire or reduce the fire. But even if you could completely put out the fire, you've set in motion a lot of other downstream aspects of this disease that are going to be very difficult to repair. So until about 10 years ago, the field really was focusing on this familial form of the disease. And yet I told you that was less than 5% of patients. So what about the other 95%? What's going on with those? Well, we knew that it was complex. Those other 95%, we knew there was evidence that all those other risk factors I mentioned influence this: aging, diet, exercise, education, et cetera. And they are modifiable risk factors except for age so far. But diet, exercise, et cetera, education, you could potentially modify those to reduce your risk of Alzheimer's. So that's the good news. But this other 95% is also influenced by genetics, but in a more complex way. So how do we know that? Well, you guys might have heard of the term twin studies. They're a type of study that's often done in medicine to try and look at the heritability, what kind of genetic component contributes to a disease. And it's quite simple. If you have an identical twin and one of them develops the disease, you can ask, what's the chance the other one develops the disease? Because they have identical genetics, right? And that tells you the overall heritability. And so here's a graph for a few different diseases, and you can see Alzheimer's right here in the middle, and that blue line is at about 60%. So if one twin has Alzheimer's, 60% chance the other will develop Alzheimer's. So that tells us our total heritability is about 60%. The good news is that other 40% we can modify with lifestyle effects. What are those genes? That's where it gets a little more complicated. Turns out that these genes are probably lots of genes having small little effects. Each one may be influencing the risk 5%, 10%, and it's only collectively that they can combine to actually lead to this 60% effect on Alzheimer's. And so for many years, we had no way to know what these genes were. you wouldn't see that heritability that you would see with the familial forms, because you might get one of those genes, but not the other 5 from your parents. So we were kind of looking for a needle in the haystack. And I guess what they figured out was, well, if you're looking for a needle in a haystack, or let's say you're looking for lots of needles in a haystack, you probably want to start with a big haystack. And so they've actually been looking now at hundreds of thousands of Alzheimer's patients and another 600,000 control patients in these studies that are called GWAS, genome-wide association studies. They're essentially looking at all the DNA of these patients, and they're trying to figure out, once you have those many numbers, what genes, what areas might influence that overall risk. And it's only as you start to get these very big numbers of data and patients, you can see this sort of increasing number shown here on the bottom, that you start to get the statistical power to detect these genes that have small effects. So you can see this list of genes is going up here as we get more and more samples. So these are what we refer to as the GWAS loci, or the GWAS genes. And this has really fueled sort of a renaissance, if you like, in Alzheimer's research. And you've noticed I haven't said much about microglia, right? Well, you're going to see that the reason I'm talking about microglia is really driven by this research. So one of the first questions you can ask, now you have these new clues, these new genes, is what are the brain cells that express the highest levels of them? Chances are those brain cells are going to be important for this disease process. And so sure enough, that's what a lot of researchers started doing. And this is an example of some of that data. This is what we call a heat map, and essentially red means very high expression, blue means very low expression. And up at the top, we've got different types of brain cells. And you can see these red lines here are all right under where it says microglia. So many of these genes are most highly expressed in microglia, about 80% of them. And yet, here is a figure of brain cells. I don't like this figure because there's this beautiful neuron, a really nice astrocyte, another neuron, a oligodendrocyte wrapping around with myelin. Here's this pathetic looking microglia. And really, they were sort of the ignored stepchild, if you like, of the brain for many, many years. But this data started making a lot of people turn their heads and start to say, well, we need to understand what they're really doing. So here's what they really look like. They're actually beautiful cells. They have these very ramified processes. And they are actually the immune cell of the brain. They're the only resident immune cell of the brain. So we have an immune system that we know a lot about. We've sadly learned a lot about it during COVID. In our brain is somewhat protected because we have something called the blood-brain barrier, but we do have an immune cell, and it's the microglia. But because our brain's protected, those microglia can do a lot of other things too. So they have been shown to influence neuronal homeostasis, so they can provide growth factors for neurons. They can regulate synaptic plasticity, the connections, the forming and the breaking apart of connections between neurons. They can clear away dead cells and debris, and they do a lot of that during early development. They also are important in turning over myelin, and myelin is that insulation that wraps around the connections between neurons and really kind of speeds up the conduction. And myelin is, for example, the substance that is affected by multiple sclerosis. And then quite recently, we found that they themselves are important for maintaining that blood-brain barrier and protecting our brain. So how can we start to study these cells and learn more about what they're doing? Well, the traditional approach for a lot of research, and including Alzheimer's research, is to turn to model organisms such as mice. And so one can actually take genes, put them into a fertilized mouse oocyte, mouse egg. You can see a tiny little thing here injecting a human gene into this mouse, and it gives rise to something called the transgenic mouse. And so we can do that, and we can use genes, for example, from those familial forms of Alzheimer's disease that have very strong effects to make mice that develop amyloid plaques. And when we look in their brains, not only do they have amyloid plaques in red here, but you see these microglia clustering around them, surrounding those plaques, just like we saw in the Alzheimer's brains. So that's good, and there's a lot of amazing research that's been done using these kind of models to try and understand how microglia are responding to plaques. And one of the first questions was, is this good or is this bad? And admittedly, a lot of the field, I would say myself included, would think, well, this is inflammation. This is an immune cell. They're inflamed. Inflammation is bad, right? Well, I'm going to show you that turns out it's not so simple. So we can use those mice, and we do use those mice, and many people use those mice. But at the end of the day, a mouse is not a human. In fact, here's the sort of tree, a family tree. So we are very distant from mice in terms of our overall genetics. And that becomes even more of a problem when you are studying genes, right? And I just told you we found all these new genes in microglia, and we want to know what they do. Well, it turns out a lot of the mouse versions of those genes are very different from the human ones. So we need a way to study humans and human microglia. So how can we do that? Well, it's tricky. I've asked lots of grad students. No one wants to let me do a brain surgery and take out their microglia. Oh well. So instead, we turn to stem cells. Right? Being in this building, learning from people like Brian and Eileen about all the amazing things you can make from a stem cell. And as we were starting this work in the early days and then continuing on, probably many of you have heard about this concept of induced pluripotent stem cells. So where you can take a skin cell and add some key genes temporarily, and it can kind of reset the clock to make what's called a pluripotent cell. And pluripotent cells, the name pluripotent means they can give rise to any cell type in the body, right? Or at least have the potential to. They can also essentially be grown and expanded and shared indefinitely. And so here at UCI, we've actually been making these from patients with Alzheimer's and controls. We've made over 200 of these lines. And one of the nice things, because it's sort of an unlimited supply, is we can send them all over the world for people to do all kinds of research related to Alzheimer's, but even other diseases. So that's useful, but then ultimately you then want to turn them into the cell type that you want to study. And so I think you can guess what cell I wanted to study. We wanted to know, could we make these into microglia, right? And I was actually kind of surprised when I first started thinking about this, looked on our typical PubMed, it's called, where we search for papers, and there was nothing there. There was no papers or any reports on how you could make this last step. Lots on this, lots on how to make these into neurons and astrocytes and oligos, all those beautiful cells I showed earlier. Nothing on the poor ignored microglia. So we just kind of went for it, and I got, somewhat lucky. That sometimes helps in science, but also a lot of hard work. In fact, I'm going to show you this slide that my former grad student, Edsel, hates the slide because it sums up his entire thesis in one slide. And it's not even that cool looking. But essentially, he figured out how to drive differentiation of iPS cells into microglia. And really what we did is follow what we know about how they develop in normal development. So they actually occur very early in development. We actually have a yolk sac ourselves, and that's where the very first blood lineage cells arise. They're called primitive hemopoietic progenitors, and they start to differentiate into an intermediate that then migrates into the central nervous system. And then the blood-brain barrier forms, kind of trapping them there, and the brain does the rest of the work. It provides the different cues to help them become microglia. And so essentially, we were able to come up with a way to do this. And more importantly, we had to actually prove that we had made microglia. So Edsel, I guess it was 2 slides here, I summed up his thesis. Edsel did a number of experiments to look at both the transcription, the genetics of these cells, as well as some of the function. They can eat things. For example, here they are eating amyloid. We can put them into brain organoids. Thank you, Brian. These were some of Brian's organoids that we added the microglia to. They can migrate towards certain cues. So they had all of the characteristics we'd expect in a microglia. And now we can start to use them to study these genes. And so our lab has done a lot of that kind of work, and I'm just going to touch on a little bit of it right now, talking about a gene called TREM2. And TREM2 was one of the risk genes that was first identified as being really only made by microglia in the brain. And patients who had mutations in this that impaired its function seem to have about a threefold increased risk of Alzheimer's. So a pretty big effect. The reason we hadn't found it sooner is it's fairly rare. So only about 1 in 100 Alzheimer's patients has this mutation, but it will increase their Alzheimer's. But we can ask, what is that mutation? What does the loss of that gene do? And we can do things like use CRISPR, which is a way to modify the DNA to make versions of these cells that lack that gene. Then do all sorts of genetic type of studies to see how if we change the function of those microglia. One of the things, for example, we found was that when we remove that TREM2, they can't eat amyloid as well. So that's probably part of the story. There's a lot more to it, though. We can also, though, what we can do in a dish only takes us so far, because ultimately we want to know how are these microglia going to interact with the plaques in the brain, right? How are they going to interact with neurons, or oligodendrocytes, all these other cells? So that led us to another thing, which is back to our ancient Greek. So some of you may have heard the term chimera. It was an ancient Greek monster that was described by Homer as being sort of part lion and part serpent and part goat, of all things. And there's all sorts of scary-looking pictures of these chimeras. But in biology, what we mean by chimera is an organism where we have some cells from another organism. And so what we're essentially talking about is transplanting some human cells into another species. So, of course, the cells we want to transplant are these microglial cells or their progenitors. And we decided to put them into a young mouse. And we chose young mice because that's when these cells normally would come into the brain, right? We had to do a few tricks. We had to use mice that couldn't reject the human cells, so they have an impaired immune system so that they can't reject those cells that are very different. But that gave us a mouse where we could now have human cells surviving in the brain. So there's a lot of green little dots here, and every one of them is a human microglia inside these mice. So I like to think, because we started with these nice, cute little young mice, that our chimeras look a little more like this. So what can we do with these chimeras? Well, we can look into the cells, see what is their morphology, what is their gene expression. And one of the things we found that's very quickly shown here is that by putting them in a mouse brain, we make them look and act just like they would look and act in a human brain, which was kind of remarkable and surprising. But essentially, the crux of this figure is that this purple and this green overlap. Well, the green are human microglia from a human brain, and the purple are these stem cell microglia taken back out of that mouse brain. We can also do live imaging of them in the brain itself. So here we're doing a trick where you use a laser to cause an injury to the area, and we see the processes of these microglia very quickly converging onto that injured area, trying to clean up the mess because that's their job. And then we can also make a version of this mouse that develops amyloid pathology. And so here, for example, we can see in white are amyloid plaques, and in green are our human microglia, and they migrate and start to surround these amyloid plaques and change their gene expression. So using this, we can sort of follow up on some of these studies we did with TREM2, for example, and we can see that here's a normal microglia responding to the plaques, and they're surrounding those plaques, like I showed you before. But here's what we found with the TREM2. We find that they're not really paying much attention to these plaques. In fact, if we quantify this, we find about half as many of these micro around the plaques. And so remember how I said, well, this is kind of inflammation, and there was this sort of assumption that this is bad? Well, loss of TREM2 increases Alzheimer's risk. So this bottom set of panels is mimicking worse risk. So that's telling us this is bad and this is good. So in this case, that is a type of inflammation we do want. And I think inflammation is not a great term. There's lots of different ways the immune system can get activated. Okay, so I'm going to summarize this, and then I'm going to pick up the pace to just talk about how we can use these cells therapeutically. So showing you that we can kind of learn a lot about Alzheimer's disease from this approach. We can make a lot of these cells. We can study these Alzheimer's risk genes. both in vitro and in vivo. And then learn things like what I showed you about TREM2 and what its role is. And you can imagine how we can start to use that to guide the development of drugs that we might be able to use to treat Alzheimer's. And so we're doing a lot of studies, as are many other collaborators around the world, using these kind of approaches. But I'm going to switch gears for the last 10 minutes or so and tell you about how we might be able to use these cells for transplantation. So you guys have no doubt heard about cell therapies from a lot of other people doing various cell therapies here in the Stem Cell Center. And you need a certain type of cell for a neurological disorder. You probably want a cell that's going to belong in the central nervous system. You might want cells that can migrate and respond appropriately to pathology. You may want cells that can make therapeutic proteins and deliver them around the brain. But ultimately, the very most important thing is you want a cell that's safe, so a cell that's not going to form a tumor. So fortunately, we've studied these cells for quite a while now, and we've done transplants into these mice many, many times, and we find that these cells are incredibly safe. They never form tumors. We've done over 1,500 transplants and never seen an example of a neoplasm with lots of different stem cell lines, long-term engraftment. So these cells seem to be intrinsically safe, and that actually makes sense. There's no human tumor that arises from a microglia, so there's something about this cell that makes it a safe cell. As I showed you, we can see different responses of these microglia to different pathologies. So here, for example, in addition to clustering around this plaque, they're changing their gene expression. They're turning on this one gene called CD9. And you can see it's really only turning on when they see these plaques. And so that gave us an idea that perhaps we could use that gene to drive delivery of a therapeutic protein. And so we thought a lot about the different therapeutic proteins and decided on a gene called neprilysin. And neprilysin is an enzyme that had been shown to cut up this beta amyloid. So John Paul, who's back here, is our CRISPR expert, engineered an iPS cell to add in this neprilysin, but did it in a clever way to have it delivered and turned on under control of that gene that only turns on when it sees a plaque. So he then took those cells, put them into some of our mice that develop amyloid, and one of the first things we saw is that it seemed to work as planned. So in red, we can see the nuclei of all our different microglia, and they're scattered around. But it's only the ones that are right next to these plaques that have turned on the CD9 and therefore turned on the neprilysin. So in other words, we're delivering that enzyme right where we need it, right to the plaques to cut up the amyloid. And so indeed, that's what it seems to do. We find that it reduces the amyloid pathology. So both the red and the blue in these stains, the sort of pink, are markers of this amyloid that are reduced when we use these cells. There's a little bit of a problem here, which is how many cells you can get into a brain is regulated by how many cells are already there. So microglia, as I said, are very safe. They don't form tumors, and that's because they control their cell numbers. They don't want too many or too little microglia. And so that presents a little bit of a challenge, where if you add microglia, for example, down here, some human microglia, but you also have mouse microglia, they kind of compete for that space. So you're limited as to how many of these cells you can get in. And this is a mouse brain. If you're ever going to develop this for humans, you can look at the size difference. That becomes a bigger problem. So we thought about how we might be able to deal with this, and one possibility was to see, could we kill off those microglia that we didn't want? Sounds a little dramatic, but it turns out that a colleague here, Kim Green, had developed an approach to do just that, and the mice seem to do okay. They can live for several months without microglia, amazingly. But if you take that drug away, the remaining microglia come back very, very quickly. And it turns out getting that timing down is very, very difficult. And so instead, what we were able to develop is an approach that could allow us to switch from the unhealthy microglia to the healthy microglia. And it's a long story, but I'll sort of just very briefly mention that what we did was to change one gene and just one amino acid in that gene to make our cells no longer die when they're exposed to these drugs. And so that allowed us to make cells that were resistant to these drugs. And what that meant was we could transplant the cells, get a few cells, then we put them on drugs, and over the course of a couple months, the brain gets filled with those human microglia now. So we're able to regulate the amount of these therapeutic cells that we can get into the brain. So with that, I think I am going to just get to the one last point here, which is when we do this, we're able to reduce that amyloid load, that last column there. And in fact, we even see evidence of these cells sort of getting right in the middle of those plaques and maybe eating them up and degrading them, which is something that's not been seen much in Alzheimer's disease, to actually clear some of these old persistent plaques. I'm going to skip that. Sorry, I went too long here. And wrap up with sort of a summary of where we're going here. So I showed you how we can start to learn from the genetics of these cells to figure out, can we drive a therapeutic protein under control of something specific, such as this CD9 promoter? I showed you very briefly that we can actually modify these cells to allow us to control how many cells we can get into the brain. And that is something that's going to be really helpful if we want to think about moving this forward clinically, to be able to get enough of these therapeutic cells into the brain. And really, it was by combining these approaches that we can see that there's this reduction in this amyloid pathologies, and I didn't have time to get into it, but also sort of a preservation of neuronal health. And then I'll end with sort of some of the fun stuff, which is just to thank all the wonderful people from my lab through the years, some of our pictures there, and also the fantastic colleagues and mentors here at UCI. And these are some of the ones who I've known for, wow, a long time, but have been very, very influential in my career. So thank you so much.
Beautiful talk, Matt. Thank you so much. Questions from the audience? You raise your hand and I'll walk with the microphone.
In the clinical trials with the drug, would they ever consider a person who has the potential for Alzheimer's but doesn't have it yet?
That is a fantastic question. So there's a lot of interest in doing exactly that, because the thinking is, can't we start this earlier, right? We know it's accumulating for 10 years. So there are some trials starting to do that. In fact, they're turning back to some of those familial cases, because there we know enough about the families that we can predict when they will get the disease. And so they're starting trials treating patients 10 years prior to when we think they'll get the disease. Those are just going, so it'll be a little while before we know, but I think there's a lot of hope that that's going to help us. But there's two parts to that story. So one is to do the trials and find out. The other is to have a drug that is very, very safe for long-term use. And I think the analogy we like to compare ourselves to are the statins, right? You got high cholesterol, you go on a statin, prevents you getting a heart attack 10 years later, right? The problem is these drugs are nowhere near as safe as statins. So we need to develop safer drugs, especially ones that can be provided for years. And so that's the other part of it. And then the third part is there's a lot of work on biomarkers trying to predict who will get Alzheimer's, looking at not just their family history, but measurements in their blood. And that's moving very fast, and there's a lot of progress there. So I have hope that we will get there.
If you do use someone like that, and they don't develop Alzheimer's, could you conclude that the drug prevented it?
Not with one person, but if we do have a trial with enough people and a placebo control, then yes, yeah. But it's going to be a long trial.
Great talk. I have a question. All of this research is pointing towards really getting rid of the amyloid plaque, but what about the tangles?
I'm so glad you asked that. And I would hope you guys would know to ask that. So tangles, and I didn't have enough time to really get into tangles, but tangles come a little bit later in the development of the disease, but they seem to be much more tightly linked to the actual cognitive impairment. So this amyloid cascade hypothesis, one of those downstream things that amyloid drives is the tangles. And then that's what really starts to injure the neurons. So that's another way to try and develop treatments that we might be able to use sooner in people who are already affected even. So we're doing experiments to use these cells to try and target tangles as well and try and either prevent their formation or reduce their formation. So using different approaches. And in fact, there's a whole nother arm of our research and many other people's research showing that how the microglia respond to the amyloid influences whether you get more or less tangles. So the microglia are probably the go-between between the amyloid and the tangles. And so I think that's probably the area I'm most excited about because I think it does give us the opportunity to intervene at a later stage.
Hiya. You mentioned anti-inflammatory or inflammatory responses in the brain could be beneficial. Do generic anti-inflammatories dull the microglia?
Good question. So because of this sort of initial assumption I alluded to, we all thought inflammation bad, right? So there were many trials of anti-inflammatories for Alzheimer's disease, none of which had any effect. So is that again because we're starting too late, or entirely possible? The problem is a lot of them are sort of affect too many things is one possibility. So the immune system has lots of different angles to it. And that's why I was kind of getting to this point that inflammation is a crude term. There's a type of activation we need. We need that response to surround plaques, but we also don't want other types of inflammation that might be detrimental. And so I think we actually need to develop better drugs to target that specifically. And there's a lot of work doing exactly that, trying to find the right drugs to fine-tune these microglia the way you want them to.
Are iPSC-derived microglia, are there a lot of significant differences between them and natural microglia?
Yeah, great question. Essentially, no, is the short answer. So there are a few differences, but if you take a microglia from a human brain and put them in a dish and compare them to iPS microglia in a dish, they're very, very similar. There's a few genes here and there that are different, but they are very similar. They behave similarly. The gene expression is similar. If you take these iPS microglia and put them in a mouse brain, they look very, very similar to human microglia that were in a human brain. So they're very similar. What I'm kind of getting at, though, is that a microglia in a brain and a microglia in a dish are quite different. So the process of taking them out of a brain and putting them in a dish and working with them in a dish changes them, at least while in a dish. But they are what we call plastic. They can go back when you put them back in the brain.
During the transition phase between the old microglia and the modified microglia, is the brain put at any immune risk?
Yeah, good question. Not that we can tell of so far, but I would imagine, if the mouse was inoculated with a meningococcal bacteria, it would be a bad thing, because when you do get a brain infection, it's the microglia's job to be there first. And then they call in the backup troops from the T cells and other cells. So the good thing is we have the blood-brain barrier that most of the time is protecting us and protecting these mice. But yet, it's probably not a good idea to go a long time without microglia. And so one of the things we're trying to do is to figure out, can we titrate this drug to very gradually replace the microglia? So we're not killing them all off and then replacing them. We're just giving a little bit of an advantage to the resistant cells.
Great job, Matt. So you mentioned the genetic factors that influence Alzheimer's, but you also mentioned that it's age associated.
Yeah.
So I was wondering, do microglia change with age? Do they become more dysfunctional? And if you replaced them with young microglia, could you reverse it?
Yeah, also fantastic question. Yes, they do change with age, and there's a lot of interest and a lot of studies on this, trying to figure out what drives that process. And they also change with chronic pathology. So a microglia that's been exposed to amyloid for 2 years is going to look different to one that hasn't. And how long it would take to reset them if you take them out of that environment, we don't really know yet. But as you know, one of the advantages to these stem cells is they're baby cells. You erase those epigenetic components of aging. And so by making an iPS cell, even if you're starting with an iPS cell from an 80-year-old, it's almost like you've got a baby microglia now. So they may intrinsically be healthier for that reason alone. But yet, it's a fascinating area, and the short answer is yes, they do age, and they become less able to do their job.
If nobody's going to ask, I'll take a chance. So you talked about iPS cells. They come from maybe 200 donors you have. How is this transplant going to work in a human? And won't their immune system just reject these cells? Are you going to make them hidden? What's your plan?
So I wish I had the right answer, but I'll tell you what we're thinking. And this is where, when Eileen introduced this, she said, I'm going to tell you about the very early stage work. And I agree with that. This is the early stage work about, is this even feasible? What Brian's asking about is the real, how are you going to treat a patient like this? If I take iPS cells made from me and put them in Brian's head, his immune system is going to reject those cells very quickly, right? That's what our immune system does. So how do I deal with that? And there's really two or three options. The ideal option, and it's actually the one we're trying to pursue pretty heavily right now, is what's called an autologous approach. So you actually take the patient's own skin or blood cells, you make their own iPS cell, you make their own microglia, and you put them back in to them, right? Every patient gets their own cell. That's the ideal scenario, right? That's challenging, expensive, et cetera, right? But technology is speeding that process up, and there are companies about to start a trial to do that for Parkinson's disease, for example. So that's the ideal approach. The other that you were sort of getting to is what they call immune blind cells. So there's been a lot of work on trying to get rid of some of the signals that cells have that the immune system recognizes to say it's foreign. And so you can make lines, and we're doing this to make lines that are quote unquote immune blind. The problem is that those genes that are manipulated are genes that are pretty important for microglial function. So of all the cells to try an immune blind approach with, microglia are going to be the last one to get it to work probably. And then the third option, as you know, is immune suppression. You give them an allogeneic, someone else's cells, but put them on immune suppression, and then in principle wean them off it. And I need to talk to you about this. I thought there was no way that was going to work. But apparently, the recent Parkinson's trial, it looks like it's working. So they did this approach, took them off immune suppressants, and they followed them for 7 or 8 months, and there's still signal that the cells is there and the patients are still improving. So, and this may come down to the fact that the brain is what we call immune privileged. It's a little bit more protected from our immune system. That, and there's something called tolerance that might occur with enough time. So I didn't think that would work at all, but at least the very first trial that I'm aware of that's really been moving that forward in the brain looks like it might be working.
And one last question I have is that you said that one issue with the current solution with the new drug is that you have to identify the effects and symptoms of Alzheimer's disease before you put it into practice. Yeah. What makes this treatment any different from other choices?
I would say it doesn't. It's not that different yet. And that's why I think, for example, I mean, this was sort of, and we often do this in science, especially early stage science, we ask the logical question and we do the logical test, and it was kind of, is this even feasible? And that's, again, why the other pathology, tau, was brought up. That's why I'm so excited about tau, because that now is something that people have only just started to develop tau, in those early stages. So you might be able to address that. And the advantage here is that what these cells let us do is deliver things throughout the brain. And so I didn't really get into this, but this blood-brain barrier I keep talking about, it protects our brain, but it also prevents a lot of drugs from getting into our brain. And so one way around that is to use these microglia sort of like a Trojan horse to deliver the drugs into the brain and around the brain.
So I thought just for entertainment, as you did such a great job answering questions and fantastic questions from the audience. Thank you guys so much for being here tonight. I was thinking about the very first scientific conference I ever went to. I am not that old. And I saw people almost come to blows over the amyloid cascade hypothesis at that time. I was new to Carl's lab at that time. And at that time, just for perspective, for some of you in the room, we didn't know that the brain had a stem cell population. And we had no idea how to make embryonic stem cells. And we had no idea that you could reprogram a somatic cell, a skin cell, to behave like a stem cell. Mm-hmm. And we certainly had no clue how to take those cells that we had reprogrammed and differentiate them into anything useful. And on top of that, we didn't know all of the other things about disease pathogenesis that Matt has shared tonight. And so maybe coming back a little bit to my starting theme of discovery drives everything. Discovery really does drive everything. And I think it can be hard sometimes to bring that message home. But Matt, you gave a master class in that tonight. So thanks so much.
Thanks so much.